everyone, and welcome to week 3 or module 3 of our class. This is again the general biology lab. I'm going to go ahead and share screen so we are on the same page as to what is due for this week. So let me go ahead and go share screen, and hopefully you all will be able to see my screen here. So I'm already on the module 3 webpage here. So this week we're going to be really concentrating on molecules here. So we did a little bit of chemistry last week. So a little bit further here, kind of more, in my opinion at least, it's kind of a little bit more of a funner lab or more fun lab. So looking at those chemistry foundations and now kind of going into the life portion of biology here. So again, looking at living things, how they're composed of cells, we're going to look at how these cells transport molecules to the membrane through osmosis, through diffusion, and why that's important in biology. So when you go to the introduction here, go ahead and click on the link. It's just going to have a couple tabs here about explaining some of the basic cell structures, not all the organelles, but some of them. If you're taking the lecture, you'll go more into the details here. Looking at cell function, why we use the process of diffusion and osmosis, what's the difference? When do we use these processes? Looking again at the scientific method, developing a research question as to why these processes happen, and then conducting a scientific research, based on some of the background information that we're learning here. So again, the overview, just a quick little blurb here about diffusion, how it crosses the concentration gradient, also about osmosis. So if you're taking the lecture portion of biology, or you have already, these are some terms that you are most likely familiar with. The readings and resources will reflect some of the chapter textbook. So chapter 1, looking at the scientific method again, looking at chapter 4, prokaryotic versus eukaryotic cells, and then chapter 5, looking more at the cell membrane, cell structure, and permeability in terms of the cell membrane and diffusion of osmosis. This is a link that you're going to be using for one of your assignments, and I'll show you this also in another area as well. So ask a biologist. And then some other helpful links here. So looking again at the scientific method, looking at the A to Z database of biology, and then citations, because you're going to be starting to work on your final assignment. So your lab, you have one lab and one part of your final project due. So it's called milestone 1. That's all that's due this week. So there's no discussions, nothing due Thursday, but the assignments are due by Sunday. So first, let's concentrate on the lab. So again, if you go to this guidelines and rubric, that's how you'll access the lab. So let me go ahead to the lab itself. So this lab is called Cell Structure and Function, really looking at eukaryotes, prokaryotes, and viruses. You'll enjoy this lab because it does have some lab questions, and then you're going to be drawing some organelles and some viruses as well too. So these are some of the materials that you need with your lab kit. Also some time. Make sure you do have enough time. So as you can see here, around 20 minutes to prep. Around, I would say about an hour, hour and a half for activity 1. Another hour for activity 2, and then another hour for activity 3. So quite a bit here. So make sure you have, of course, don't start this assignment Sunday evening, 11:30 at night, because you're really not going to have time to do this. So make sure you get started on it today. All right, so these are, I'm highlighting here. Hopefully you can see this on the screen as well. These are some additional materials that you'll need. Nothing too significant. You can be working with some potatoes for your experiment here, some spoons, tap water, of course, paper to draw on, a cutting board, a timing device, and labeling tape as well. So I'm just kind of scrolling over what should be. Again, making sure that you all you sign this lab safety agreement, so making sure that you use gloves, an apron, goggles, of course. You're not going to be working with any dangerous chemicals or anything, but just want to make sure that you're being safe. So make sure you have closed-toed shoes, so don't do anything barefoot, just in case you drop anything. We don't want to cut our feet or anything. A little bit of a background here on the topic. So looking at cell membranes, what does this term here, meaning semi-permeable, what does that mean? Some things are allowed to pass through, some things are not. Why? Why is this important? You know, think of viruses. If every single virus that you have come across was able to infect your cells, that would not be a good thing. You'd be sick a lot or worse. So what does that selective permeability do? Why is it a good thing? And why is it not a good thing? You know, it can be a not so good thing as well too. What does this mean in terms of surface to volume ratio? So our cells grow as you age, as you go through the stages of life, from a baby, an infant, to a child, to a young adult, to elderly. So why is that important to know the surface to volume ratio? You know, why is this important to cells like neurons? Why is it important to other types of cells? What does that mean in terms of gaseous and wastes and things like that that come in and out of the cell? So again, more background here on osmotic pressure, looking at osmosis. You can come across these two terms. I'll go back a slide in terms of tonicity. So hypertonic versus hypotonic versus isotonic. What do those terms mean? Why are they important? What are they use? Like why do we use them in biology? So you'll be coming across that with your lab. And I'm going to stop here at the knowledge check because then you need to proceed there with the rest of the lab here. All right, so let's go back to milestone 1 here, or milestone 3 here. Okay, so that's the lab. And then this is going to be your final project. So you're going to turn in milestone 1 of your final project. And this is basically going to be your research topic. So you're going to formulate a research question that you're going to explore throughout the term. There's a link here. If you go to the guidelines and rubrics, I kind of did a shortcut here, and you go to the Ask a Biologist. Remember that link that I showed you previously here? So there's various topics that you can choose. You can talk about air pollution. There's one about an ant farm here, birds and their songs. I don't think anybody in my classes have ever done this one. Might be a little bit tough to do, but birds and their songs. Composting is one as well too, and there's several others, so just click around here. So you are going to, here's the directions for the milestone 1. You're going to submit your research question and a little bit of background information. So make sure that the topic that you choose, and again, you don't have to choose one from that Ask a Biologist. They're just suggested for you. Just be sure that whatever topic you use, you're going to be able to complete or answer your hypothesis using the scientific method. So you're going to formulate a research question that you want to explore and then provide a little background information for this as well. So here's just a few again. When water gets icy, there's one about doing the Manduka caterpillar as well too. So here's what you're going to submit. You're going to do a research question, and then why is this significant? So make sure you answer these 2 questions. How this experiment relates to the significance of biology or the basic biology principles, and also how might this information answers give you some information in terms of conducting your experiment in the field of biology. So use the rubric when you are submitting in terms of how I will be grading. So use that, and I believe that is about it. So again, what's due this week? Your cell structure and function lab due by Sunday. Make sure you complete it on the online Carolina Biology platform. Download it, submit it into Brightspace here. Your research 1 questions, you're not going to use the biology lab for that one, the online one. You're just going to use the template here. I believe there's a template, or if not, you're just answering those questions off the rubrics and guidelines. So just submit this. And then here's just a quick little reminder for lab 4. Or module 4 lab. That one's going to take a while. So make sure you plan your time accordingly. So I would just definitely recommend reading up on that one. After you complete everything for this week, read up on that one and make sure you just kind of plan ahead in terms of time management. No discussions or anything this week. So again, just these 2 assignments. Let me know if you have any questions, and look forward to a great week this week.